Good afternoon, as we say in Wales. First of all, thank you for allowing me to speak at this conference today, where I'm going to talk about the uncoupling of synaptic loss from amyloid burden in an Alzheimer's disease mouse model that has a protective variant of PLC gamma 2. So I'm part of the UK DRI at Cardiff, and I want to start with a little bit of a background on PLC gamma 2. So research led at Cardiff by Julie Williams and Rebecca Sims identified rare coding variants in a gene PLCG2. So in 2017, this was identified to decrease the risk of developing late onset Alzheimer's, and is P522R. Since this discovery, others have replicated this finding, not only in Alzheimer's disease, but in other dementias as well. And it's also been found to increase the likelihood of longevity in cases older than 100 years. And I would just like to highlight that this P522R variant was present in the latest GWAS study for AD. So in the brain, PLCG2 is primarily expressed by the microglia. And I've just lifted this from this fantastic paper, which highlights that PLCG2 encodes an important enzyme, so it's sitting here, that is downstream of our favorite receptor, TREM2, and is involved in the hydrolysis of lipids, modulating intracellular calcium and some signaling pathways. It's important to note that the PLC gamma 2 enzyme activity controls microglial motility, it alters phagocytosis, survival, and proliferation. And the most important thing, it is a drug target as it is an enzyme. So the research that we had the hypothesis that the PLCG2 P522R variant confers some sort of protection against pathology in an APP mouse model. So my work and background is mainly histology and microscopy. So our first sort of aim was to sort of identify what influence does this variant have in the wild-type brain, and then does this variant alter the APP pathology in the APP NLGF mouse model. So I don't know whether if you can see this in the bottom corner, but we, in the lab, we made a PLCG2 R522 knock-in model with the Jackson Labs, which is currently available. And for consistency throughout the talk, everything colored in blue is the wild type or the risk variant, so P522, and everything in orange is the protective variant, R522. All this data is currently unpublished. It is hot off the lab. We have submitted it, so I hope you like it. If you don't worry. It's all from 6-month-old mice, equally weighted, male and female, with an N of 10 for all groups. So, okay, all in the hippocampus CA1 region as well. So we first started at looking at the microglia distribution in the tilin, so we stained it with IBA1 in the hippocampus, and we found that in the protective variants, this is in the wild-type mice, we found that the R522 protective variant actually increased the density of the microglial coverage in the hippocampus. So we decided to look at the microglia a little bit closer, to look at their morphology, obviously remembering the given context of time and the environment and location and age. So we've got the Imaris tracings, which I think everyone has seen throughout the talks. And when we look at the microglia from the R522 mice, the microglia actually show reduced ramifications, reduced terminal points, and also have a reduced cell volume as well. So it looks like the microglia in the wild-type mice with the protective variant have got some changes. So we co-stained the microglia with CD68, so a lysosomal marker, reconstructed again in Imaris, And we found that the amount of CD68 relative to the IBA1 coverage was actually increased. So the R522 in the wild-type mice has an increased prevalence of the CD68 lysosomal marker in the microglia. So since we have an effect in the wild-type mice, we decided what happens in the context of APP. So I'm not going to go through the model too much, but we use an APP NLGF mouse model, which is a knock-in model for amyloidosis. So it doesn't have any overexpression artifacts like the other models that have. And at 2 months of age, the mice develop plaques, and by 6 to 7 months, the plaques are almost near saturation. So we can stain the hippocampus with thioflavin S, which is a plaque dye, and you can see these sort of hot spots of where the plaques are. And then we stained for the plaques in the R522, and we found that the protective variant increased the plaque coverage in the hippocampus. So we've got a protective variant, but with a mouse model with more plaques. So we were like, okay, what's going on? So we've got different measures for the plaque. So we had an area coverage was increased, and also the number. So we have an effect in the APP mouse with this protective variant. So we decided to follow this up with the microglial counts, co-staining with an amyloid dye. And we found that the R522 protective variant increased the global microgliosis in the hippocampus of the APP mice. So we've got an increased plaque burden and an increased microglia coverage with a protective variant for Alzheimer's. So we decided to look at the periplaque region to try and understand what was going on in a little bit more detail. So we can co-stain, as I showed before, with the plaque stain, the microglia, and the CD68, and we can do a nice 3D reconstruction to see where the immunoreactivity is. And I hope you can appreciate that you can see that there's less microglial response to the plaque, and this also coincided with less CD68. So in the context of the periplaque region, the protective variant, the plaques don't seem to have normal IBA1 response or CD68 response. So we know that the APP mice have a synaptic phenotype, where you age the mice, they have synaptic loss. It's quite evident at the plaque. So we quantified these periplaque regions for the plaque stains, microglia, CD68, and PSD95, which I hope you can see on there, which is a postsynaptic marker for the synapses. And then we recorded any of the engulfed PSD95 that was located within this periplaque region. And quite surprisingly, we found that the protective variant coincided with a reduced engulfment of the synaptic puncta, and with a higher periplaque synaptic puncta around the plaque area. So it looks like we have a reduced synaptic engulfment at the plaque, but what happens sort of outside of the plaque further away? So in the wild-type mice, we stain for the synaptic puncta for bassoon and PSD95. So this is a pre and post synapse, and any white hotspot you see here is a colocalized synapse. So in the wild-type mice, there's not much visual difference, but I hope you can appreciate in the NLGF Alzheimer's mouse, you can see there's a loss of this synaptic puncta. But in the Alzheimer's mice, sorry, I shouldn't call them Alzheimer's mice, the amyloid mice, we had the R522 variant. We actually found that this synaptic puncta were actually protected from this synaptic loss in the APP mouse. So it looks like we have an R522 protective effect on the synaptic density. So in the lab at Cardiff, I have developed a technique which you can label neurons and highlight the synaptic density in another approach through the dendritic spines, looking at the dendritic morphology. So we use this using a dye called dye I, and it randomly labels neurons, and it injects into sort of single neurons at a time, and you can see synapses at a really good resolution. So in the wild-type mice with the two variants, visually there isn't much difference, But then in the Alzheimer's mice, you can see that there's starting to be some synaptic loss here. And this is what you would have seen from the synaptic puncture data. And I think this is my last bit of data. So we have the APP mice with the R522 variant. We looked at the synaptic density of the synapses in the hippocampus, and we were able to show that the synaptic density was increased and protected in the APP mice with the protective variant. And we can separate the dendritic spine protrusions into different subtypes based on their morphology, so the size of the head and the neck of the synapse. And we can separate them into stubby, mushroom, and thin. And we can see that we've got a protective effect of the R522 variant in all three subtypes of the dendritic spines. So we have the protective variant protecting hippocampal spine density in the APP mouse. So I'd just like to summarize. So the PLCG2 gene with a variant P522R decreases the risk of developing late onset Alzheimer's. The activity of the enzyme alters microglial function. And the data that we found, the ABP mouse model of amyloidosis with the protective variant increases plaque burden in the hippocampus, increases hippocampal microgliosis. However, on closer inspection, the periplaque microgliosis and CD68 response is reduced, but we have a synaptic effect with the protective variant, which might be the effect that we see in the human case as well. So I'd just like to say thank you all for listening. These are all the guys that have helped with the work, specifically my supervisor, Phil, and Julie, who secured the funding for this work. And all my links can be found here. If you have any questions, I'm more than happy to take them, or just find me in the break next. Thank you.
Yes. Thank you, Ryan. Questions? Yeah, down here, there's two. Oh, up there, yeah.
Just in terms of, you said that there was an increased sort of total area of the plaque, but did you see sort of subtle differences, like a difference in diffuse plaque versus maybe sort of more dense core plaque that could arguably, people say, is more inert?
Yeah, so we haven't stained that yet, but that is something that we would like to consider. So we've just done that based off the thioflavin S at the moment.
Yes, down here, yes.
Thanks, Ryan. It's a fantastic talk. Very interesting work on PLC gamma 2. So we've been looking into this using IPS microglia co-culture systems with one of my students. So just for full disclosure, those were healthy donor lines that we CRISPR edited, right? So I think your studies nicely show this in the importance of looking on a relevant background. But I wondered if you, just to clarify, your mice, they're homozygous for the variant.
They are homozygous.
Right. So
yes.
did you also look at heterozygous?
No, I haven't looked at heterozygous.
Because I wonder, really, the question then is there like a sweet spot for PLC?
Yeah. So there's several studies that have shown so the variant causes hyperactivity of the enzyme, but that activity actually depletes its substrate, so it's not exactly as clean as what people may think. But yeah, we haven't looked at the het. I did propose that, but I don't think it would have fitted in properly with time.
Yeah, because I think just anecdotally, well, not anecdotally, we showed it, but in vitro at least, the synaptic effect doesn't seem to be so much dependent on that. So we see the same preservation of synapses. But the uptake of at least exogenously added amyloid to those cultures is higher in the heterozygous than the hom.
No, definitely something to consider.
Thanks.
For that. Thanks.
So up here.
Hello. Thank you for your great presentation. I want to ask you, how did you load synapses with the DII?
So I've got a technique called diolistic labeling. So it's a particle delivery system where it randomly fires fluorescent bullets onto a tissue slice.
Okay. And the other question is about the synaptic activity, because you show that there is an alteration in synaptic density and also the synaptic morphology.
Yeah.
So I was wondering if there are some data on literature about the synaptic activity or synaptic transmission in this animal model.
Yes, so the functional activity of the synapses wasn't part of the project, but it is definitely something that we're following up with now.
So your microglia with your protective allele, if you RNA-seq them, do they have this typical amyloid-associated gene signature?
Yeah, so we're starting with the amyloid-seq now.
Ah, okay.
So I think that'll come through in the next couple of months, hopefully.
All right, thanks.
I was just wondering, you talked about it being primarily a microglia in the brain. Do other cells in the brain also express it, and is it anywhere comparable to microglia at all? Or?
Yes, so it's primarily expressed by the microglia, but the neurons of the dentate gyrus do have some low expression, but it's primarily the microglia.
Thank you. Thanks. Thank you, Ryan.
Thank you.
Thank you.